In this video we will understand that late stage activation of M1-like macrophages that is correlates with the immune checkpoint therapy response. So how they come up to this point? So their initial motivation was to understand more about this late stage activation of M1-like macrophages. So they compared the single cell RNA-seq data and of these monocytes and macrophages and are able to cluster them into 10 different populations based on the top differentially expressed genes, and that cluster is shown here in the Disney plot. And the heat map also shows that differential expression of genes leads to the 10 different clusters. And what are the genes that are expressed in different populations are also shown here, and based on the gene expressions they have classified those populations or those clusters into different categories that are shown in this panel C. So, and among these clusters, the important thing that they have find out is that this late stage M1-like macrophages genes are upregulated in this cluster C, and from the pseudotime, what they have find out, pseudotime analysis, what they have find out is that this populations occurs late. These populations occurs late compared to the other clusters. So far their analysis seems to be in line with their hypothesis that late stage M1-like macrophages, this M1-like macrophages cause late stage activations. And after that what they did is that they checked the other pathways using GSE analysis. They checked the pathways that are dysregulated in this cluster 3, and what they have find out is that most of the cell killing pathways and the motility of the cells are upregulated, such as these leukocyte migrations, the cell killing, this oxidoreductase activity, the inflammation such as inflammatory response are upregulated compared to the other clusters. And once they identified that it's the cluster 3 that resembles with the late stage activation of M1-like macrophages, and from literatures what they have find out is that similar kind of response are observed, or similar kind of genes are dysregulated when the patients are treated with immune checkpoint therapies. Even patients are treated with immune checkpoint therapies. So in order to validate that analysis, what they did is that they take the data of the patients who are treated with this mono immune checkpoint therapies, so that is anti-PD-L1 antibodies, so that the immune system will not be inhibited by this checkpoint inhibitors that are present on the tumor cells. So patients who are gone under those mono anti-PD-L1 therapies, their data has been taken, and then they compare the dysregulated genes in those patients with the dysregulated genes in the cluster. And what they have find out is that in the 72% of the those dysregulated genes are overlapped with the cluster 3. So this further substantiate their hypothesis that the immune checkpoint therapy cause the similar response, or it will also cause these late phase activations of M1-like macrophages as they have observed in their models. And thereafter, once they have identified this similarity, they decided to check whether they decided to check whether this vaccine treatment that has been used for their model can be combined with this immune checkpoint therapies, so that it will increase the survival durations of the patients. So they did the experiment on the mice model, and what they have find out is that when they use only anti-PD-L1 receptors, they see some increase in the survival rate, that is 2 out of 10 mice are able to survive. However, when they use only the vaccine, they only 3 out of 21 are able to survive. However, when they use vaccine and anti-PD-L1, they can see that around 6 out of 20 are able to survive. So the percentage is high when both the vaccines and anti-PD-L1 therapies are used. So indicates that the vaccines can sensitize the immune checkpoint that are present can sensitize the immune checkpoint that are present on the immune cells. And once they identified that this these vaccines treatment can be combined with this anti-PD-L1 therapies to increase the efficiency of the overall treatment. Thereafter, they wanted to check whether the CT signatures are present in the patients or not. Specifically, they check the sequencing analysis, and what they have find out in the RNA sequencing analysis is that of the these mice models, what they have find out is that although they find out that only small portions of the cells are can lead to this M1-like late stage activation of M1-like macrophages. However, uh, when they check this C3 signature scores, they are able to find out that in the responsive, that is in the mice that respond to their therapies has high C3 signature scores compared to the case where mice does not respond to their vaccines plus anti-PD-L1 therapies. This indicates that all the responsive responsive treatment will cause, or all the treatment that respond to the vaccine and anti-PD-L1 will cause late stage activation of M1-like macrophages. So from this results, they finally conclude that the late stage M1-like macrophages is also occurs in the patients who are treated with immune checkpoint therapies such as anti-PD-L1 therapy. So this is not a specific phenomena only to the pancreatic tumors, but it is also this phenomena late stage activation of M1-like macrophages is also available in other tumors also that they have shown using this anti-PD-L1 therapy data. So I hope you understood how they come up to their conclusion, and if you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.